Good morning, student. I am Dr. Nikunj Patel from Department of Biochemistry, Smimel Medical College, Surat. In today's lecture, I will talk about the digestion of protein and absorption of amino acid. You have already learned about the protein chemistry. Protein, it is a polymer of amino acid linked by peptide bond. Major source of the nitrogen in our diet, it is come from the protein. Protein itself, it is a complex molecule. It is organized in a tertiary or quaternary structure. So it is too large to absorb from the intestinal mucosal cell. So it must be digested first, converted into the smaller compound like dipeptide, tripeptide or individual amino acid, and then it is absorbed from the intestinal lumen. So remember, the protein must be digested before its absorption. So for digestion of the protein, we require the various chemical and enzymatic molecule. Some of the exception for the that rules for digestion, it is in case of the newborn baby. In case of the newborn baby, that immunoglobulin that is come from the milk, okay, mother's milk, it can be directly absorbed without digestion. But in majority of the cases, protein must be digested before it absorbed. So for that, we require the various enzymatic molecule that is known as a proteolytic enzyme. So what is the meaning of digestion? Digestion, it is ultimately convert the complex molecule into simple compound. In case of the protein itself, it is a complex molecule converted into the amino acid or dipeptide. It is converted into the smaller compound. For that, we require the various proteolytic enzyme. In case of the protein, the proteolytic enzyme are secreted in the inactive form. That is known as a zymogen. Why it is secreted in the inactive form? Okay. To prevent the autodigestion of the gland from which it is secreted, it secreted in the inactive form. Like various pancreatic enzymes are secreted in the inactive form to prevent the autodigestion of the pancreatic gland. Same like pepsinogen, it is secreted from the chip cell of gastric mucosa to prevent the mucosal damage. So after secretion in the lumen, it converts into the active form with various mechanisms. So remember, for the digestion of the protein, the various proteolytic enzymes are required. Majority of these are secreted as an inactive form. In general, the proteolytic enzyme are divided into two groups depend on where it is act on peptide bond, endopeptidase and exopeptidase. Endo, name itself suggests it is act inside the polypeptide chain. Exopeptidase means it is act on the end of polypeptide chain. Suppose like that is a polypeptide chain of one protein. When it is open, okay, so they are, and that is a N terminal and this one is a C terminal. So enzyme act here and N terminal and C terminal, it is known as a exopeptidase. For the N terminal, it is known as a aminopeptidase. For the C terminal, it is known as a carboxypeptidase. An enzyme that is act inside the polypeptide chain, it is known as a endopeptidase. So various example of the endopeptidase include pepsin, trypsin, elastase, chymotrypsin. Various pancreatic and gastric mucosal enzyme are the example of the endopeptidase. So they act inside the polypeptide chain. Overall, both exopeptidase and endopeptidase act on the peptide bond. It is utilized to break the peptide bond, but location of the peptide bond are different. So overall, major three organs are the important for the digestion of the protein that ultimately release the various enzyme, proteolytic enzyme, and helps in digestion of protein. First, gastric mucosa, second, pancreas, and third, intestinal mucosa. From the gastric mucosa, enzyme secreted, it is mainly pepsin, and in case of the children, it is renin. Second, pancreas, it secretes the pancreatic juice that contain the various proteolytic enzyme include trypsin, chymotrypsin, elastase, and procarboxypeptidase. And from the intestinal mucosa, the enterokinase, dipeptidase, tripeptidase, and proaminopeptidase. And the various enzyme helps in digestion of the protein. So first digestion are the start in the stomach in case of the protein. The saliva does not contain any type of the proteolytic enzyme. So directly first digestion of the protein are the occur in the stomach. In the stomach, the main enzyme are the required for the digestion of the protein. It is pepsin. In case of the children, it is a renin, and chemical compound HCl, hydrochloric acid. It is also helps in a digestion of the protein. It does not directly hydrolyze the peptide bond, but it denatures the protein. So that ultimately helps to pepsin to act on the protein easily. It converts the pepsinogen into the pepsin. Okay. So it is required for the activation of pepsin. It kills the bacteria and maintain the pH around 2. It is base pH for action of the pepsin. So hydrochloric acid itself, it does not hydrolyze the bond, but it helps the pepsin to digest the protein. Second, it is a renin that mainly secreted in case of the children. It is absent in the adult. It is required for the milk protein digestion. The main protein in the milk, it is a casein. So casein, it is converted into the paracasein, and then paracasein, it is further digested by the pepsin. So remember that renin, it is a differ from the renin. It is secreted from kidney and maintain the blood pressure. So that is a hormone, and that renin, it is a enzyme. So digestion of the protein in the stomach are mainly by pepsin, that is helped by the hydrochloric acid, and in case of the children, it is by pepsin as well as renin. Pepsin, it is main enzyme for the digestion of protein in the stomach. It is secreted as an inactive form zymogen in the form of pepsinogen, secreted from the chip cell of gastric mucosa. HCl, it is converted into the pepsin by removing the around 44 amino acid from N terminal of polypeptide chain. So it converts the pepsinogen into the pepsin, and it act as a endopeptidase. it is specific for peptide bond that is formed by carboxyl of aromatic amino acid, mainly tryptophan, tyrosine, phenylalanine and methionine. Okay. Suppose that R1 if R1 that carboxyl group it is dominated by this amino acid, only in that case that pepsin it is act on this peptide bond and breaking that peptide bond. Okay, mainly aromatic amino acid, tryptophan, tyrosine, phenylalanine along with that methionine. So it is a bond specific enzyme, bond specific endopeptidase. Second major digestion of the protein are the occur in the pancreatic juice. Okay, in the duodenum. Pancreatic juice it is secreted by pancreas through the pancreatic duct. It enter into the duodenum. It contains the various proteolytic enzyme along with the enzyme for the lipid digestion. They are secreted as a inactive form. Optimum pH for pancreatic enzyme it is 8. It is mainly maintained by the bile and pancreatic juice. They are always secreted as a zymosan. And various hormone that is help in a secretion of pancreatic juice include cholecystokinin and pancreozymin. When food bolus are enter into this duodenum, it produces the various reflex that ultimately release the various hormone that act on the pancreas and release the pancreatic juice. Through pancreatic juice, various proteolytic and lipid digestive enzyme are enter into the small intestine where it is act on the food and leads to the further digestion. Major enzyme present in the pancreatic juice for the protein digestion, it is trypsin. It is secreted as a trypsinogen as an inactive form, converted into the trypsin by another enzyme that is known as a enterokinase or peptidase, enteropeptidase. That is secreted from the intestinal mucosa. From the N-terminal, certain polypeptide chain it is removed and it converts the trypsinogen into the trypsin. Then trypsin, it is also act as an autocatalytic molecule, means it further activate the more trypsinogen into the more trypsin molecule. It also convert the chymotrypsinogen into chymotrypsin, pro-carboxypeptidase into carboxypeptidase, pro-elastase into the elastase. So remember, when trypsin it is formed, it further activate the conversion of the inactive enzyme molecule into active enzyme molecule. Optimum pH for all the pancreatic juice enzyme it is 8. It is also act as an endopeptidase. It is also bond specific enzyme. In case of the trypsin, it is mainly act on the arginine and lysine peptide bond. When peptide bond it is formed by carboxyl group of arginine and lysine, it is mainly digested by trypsin enzyme. Same like chymotrypsinogen, pro-carboxypeptidase and pro-elastase also bond specific endopeptidase. Like trypsin, it is for the arginine and lysine. You can remember as a TAL, TAL, trypsin for the arginine and lysine. Chymotrypsin, it is more or less same like the pepsin. All the aromatic amino acid plus methionine are the present. In case of the elastase, you can remember as a gas molecule, glycine, alanine and serine when involved in the peptide bond formation. Pro-carboxypeptidase A You can remember A for avyl, alanine, valine, isoleucine and leucine. Okay, you can remember as a avyl, alanine, valine, isoleucine and leucine. And B, it is stand for same like the trypsin, arginine and lysine. So overall, all the pancreatic enzyme are the bond specific endopeptidase. They break the peptide bond. It is formed by the specific amino acid. Further digestion occur in the small intestine with the help of the another enzyme that is secreted by the intestinal mucosal cell into the juice of intestinal juice that include dipeptidase and tripeptidase. They breaking the di and tripeptide into the individual amino acid that can be further absorbed from the lumen. So overall, mainly three organs are involved in the digestion of the protein: stomach, gastric juice, pancreatic juice, and intestinal juice. Now, after conversion into the smaller compound like amino acid, dipeptide or tripeptide, they are absorbed from the intestinal lumen into the mucosal cell, and then it is entered into the blood circulation, mainly portal circulation, and enter into the liver where it is further metabolized. Some of the amino acid like branch chain amino acid, it can directly go from the liver to the muscle for the further utilization. Major absorption of the amino acid from intestinal lumen, it is depend on the sodium dependent secondary active transport mechanism, mainly from the apical membrane from lumen to the mucosal cell, that is mainly depend on the secondary active transport mechanism. In the basolateral membrane, that is mainly facilitated transport mechanism, that is sodium independent facilitated transport mechanism. And some of the amino acid mainly in the renal tubule are reabsorbed with the help of the Mr. cycle, that is also known as a gamma glutamyl cycle. Sodium dependent secondary active transport mechanism means it is depend on the energy, but that pump it is required for the absorption of the amino acid does not directly utilize the ATP, but independently it is depend on the ATP. That pump it is required for the absorption of the amino acid. It utilizes the sodium gradient for absorption of the amino acid from lumen to the cell, but it does not utilize the energy, remember, but it requires the sodium gradient. That gradient it is mainly maintained by the another pump that is known as the sodium potassium ATPase pump. That pump utilizes the ATP to maintain the gradient. That gradient later on it is utilized by the pump. So amino acid are the absorbed. Same mechanism are the utilized for the glucose absorption from the intestinal lumen. Here in that case sodium glucose are the use the same common transporter mechanism. That principle can be used in the formation of the ORS. Okay, super ORS means sometimes ORS contain the amino acid that help in a quick digestion, quick absorption of the sodium from the intestinal lumen to maintain the electrolyte and water balance. Because when sodium are the absorbed, along with that water molecule are the passively absorbed from the intestinal lumen. So maintain the electrolyte and water balance in case of the dehydration. Same principle are the utilized for the formation of the ORS. So that is a sodium dependent secondary active transport mechanism. It is mainly required in the apical membrane absorption. Various transporter that is depend on the secondary active transporter are the common for the certain amino acid. That can be group like neutral amino acid utilize the common transporter, basic amino acid utilize the common transporter, acidic amino acid utilize the common transporter, amino acid are the utilize the common transporter, and the last one it is beta amino acid. Some more group can be identified. So overall in case of the neutral amino acid like avid, alanine, valine, isoleucine and leucine, along with that aromatic amino acid, phenylalanine, tryptophan and tyrosine are they absorbed with the help of their common transporter, neutral amino acid. Okay? Alanine, valine, isoleucine and leucine, same like for the carboxypeptidase A. Okay? That this amino acid are the absorbed by that common transporter mechanism. Protein. Second, basic amino acid like mainly arginine and lysine along with that cysteine and ornithine are the utilize the common transport protein. Acidic amino acid like glutamic acid and the aspartic acid are utilized the common transport protein. Amino acid, it is a proline plus glycine plus hydroxyproline utilize the common transport protein, and beta amino acid like beta alanine utilize the common transport protein. So various protein are the available for the transport of the amino acid from intestinal lumen to the mucosal cell. They can be shared by the some group of the amino acid. So depend on that, various group are identified. From the mucosal cell to the portal circulation, that mainly amino acid are the transport by the facilitated transport mechanism. Okay, by ping pong model, the amino acid are the absorbed. It is bind with the protein, transport protein, and some of the conformation change occur in the transport protein, and it is open on other side of the membrane and release that amino acid into the portal circulation. So it is energy independent transport mechanism. Third, for the mainly in case of the renal tubule, for the reabsorption of the amino acid, it is known as the gamma glutamyl cycle. It utilizes the 3 ATP for the absorption of the single amino acid. Okay? Glutathione are the play important role in this cycle. Okay? Another purpose of that cycle, it is regeneration of the glutathione. Also, okay? Glutathione, it is a tripeptide. It is mainly made up of glutamic acid, glycine and cysteine. So when amino acid, it is transport by glutathione, the glycine and cysteine it is removed and amino acid are the absorbed. After that, sequentially that cysteine and glycine molecule are the incorporated into the glutathione and produce the glutamyl cysteine and finally glutathione. So that by regeneration of the glutathione, it utilizes the 3 ATP. So it is very less process for the absorption of the amino acid, that is mainly for the glutathione regeneration. So that is all about the absorption of the amino acid from intestinal lumen to the mucosal cell and reabsorption of the amino acid from renal tubule. Remember that same mechanism also utilized by the kidney for reabsorption. Whatever amino acid is filtered by the glomeruli, it is entered into the renal tubule. From the renal tubule, it can reabsorb by same mechanism. So for the absorption from the intestinal lumen and reabsorption from the renal tubule, both are sharing the same mechanism for absorption of amino acid. Certain disorder related to that digestion and absorption of the protein include disease of the pancreas like chronic pancreatitis, accidental removal or surgical removal of the pancreas, injury to the pancreas that ultimately affect the pancreatic cell. So pancreatic juice are not formed in proper amount and does not enter into the intestinal lumen. So digestion of the protein along with the lipid are the impaired. So lipid, undigested lipid are the enter into the stool, and the condition it is known as a steatorrhea. So fat soluble vitamin are the not absorbed in that case mainly. Certain small intestinal disease, mainly autoimmune disorder like celiac disease, also affect the absorption of the amino acid from the intestinal lumen. Okay, various inflammatory disorder of the intestinal wall that ultimately affect the absorption of the amino acid from intestinal lumen. Whereas genetic disorder related to the absorption of the amino acid from renal tubule include Hartnum disease, glycinuria and cystinuria. In that case, what happened that transport protein are the impaired by the mutation. Like in case of the Hartnum disease, transporter for the tryptophan are affected. So tryptophan are the not reabsorbed. So loss in urine. So less NAD form. So niacin deficiency are the seen. Leads to the pellagra-like symptoms. Diarrhea, dementia and dermatitis. So Hartnum disease, it is a defect of the tryptophan transporter in kidney. So tryptophan are not reabsorbed, loss in the urine gives the niacin deficiency and pellagra-like sign and symptoms, diarrhea, dermatitis and dementia. Same like another transport mechanism like the glycine transport are the defect leads to the glycine are the loss in the urine gives the glycinuria. For the basic amino acid, that is for the coal, okay, cysteine, ornithine, arginine and lysine, the transporter when affected, cysteine it is a loss in the urine gives the cystinuria. In that case, what happened that cysteine it is a precipitated in tubule and gives the stone. The person are more likely to be affected in that case by renal tubular stone. So basic treatment in case of the cystinuria, it is a hydration. Okay, one of the modality of treatment it is a hydration. So that ultimately prevent the cysteine precipitation in the renal tubule. So there are various disorders related to the protein digestion and the absorption. If any query related to the protein digestion and absorption, you can ask me. Thank you.